Okay. I'd like to welcome everybody to the fourth and final lecture in today's series. My name is John Golbeck. I'm a faculty member at Penn State. Over the next 4 days, we're going to be introduced to a lot of different instrumental techniques. But it's equally important that we also look at theory. And every one of us probably deals with oxidation reduction reactions, dealing with enzymes. So electron transfer theory is part of what we need to know. Now, 2 years ago, when we ran the symposium, I gave this lecture, but I ended up, in prepping for it, pulling from a lot of Les Dutton's work. And so I thought it was only fitting that we would ask Les to come today and to give the talk and tell the story correctly. So we're looking forward to an exciting talk, Les, and it's all yours.
Well, I'm looking forward to an exciting talk too, but who knows? Right, so that's myoglobin. I always have it at the beginning of my talks. I don't know why. But right, I'm Les Dutton. I'm from the University of Pennsylvania. And I'm going to talk about biological electron transfer. And I'll start out by making 3 assertions, and then I'll tell you what I'm going to do about them. So the first one you may agree with very easily, that in biology, catalytic physics and chemistry of light harvesting, charge separation, photochemical charge separation, and oxidative and reductive energy conversion, metabolism, owes everything to the control of electron transfer. Without that, nothing. Something that some people don't agree with is that natural energy systems have evolved an adequate common engineering for electron transfer in energy conversion and catalysis. It's even simple. So as I talk you through the theory, it is one of these exercises I've been working on for 30 years that as time has gone by has become simpler and easier to understand. Which brings me, and I should say parenthetically, some people think it's not quite so simple. I should say that. But nevertheless, the proof of the pudding is in the making. And I'm asserting that this learned engineering and the theory behind it, retooled for biology, is readily applicable for exploring uncharacterized oxidoreductases, of which there are many. And key to all this is to consider the construction of man-made systems that match or exceed natural ones that inspire them. So I'm not going to come out with the Feynman quote about understanding and making, but that is the essence of this. So if we do understand the theory enough, then, and we can make something from scratch, no copying nature, then we've succeeded. So what I'm going to tell you about is first what's needed to describe, measure, and understand biological electron tunneling. It is quantum mechanical, and it's coupled events, chemistry. And that's what gives rise to electron transfer. But the underpinning action is always tunneling. So I'm going to identify which of these many parameters in physics and chemistry actually, in a Darwinian sense, have been selected to make biological enzymes and electron transfer systems. And I'll show you at the end that these naturally selected parameters by nature that I'll tell you about, they're understood sufficiently to consider the creation of man-made oxidoreductases for function not just in vitro, but in vivo, for the benefit of mankind, and in other words, in the clinics, and to get these oxidoreductases into the cells, into the mitochondria, to repair genetic lesions. That is the goal of the beginning of this work, understanding the theory behind electron transfer. So I'll give you some statistics first. You all know this biochemical map. Never look at it, I'm sure. But in there are many oxidoreductases. So of the enzymes that are in that map, 1/4 are oxidoreductases. There's probably more, but it's a large number. And they're comprised of dehydrogenases that transfer not electrons, and I'm not going to go too far with the hydrides, but 2 electrons and a proton together as a unit. These don't travel very far. They do tunnel a bit, apparently. Electron transfer coupled to protons separately, often, 4%. These enzymes cover all the gaseous enzymes and small ionic enzymes. They are the first enzymes to receive the sunlight on Earth. And they produce oxygen, mop up CO2, and deal with nitrogen exchange in the Earth. They are critical. And then there's some that are both. They appear here and there everywhere, but often have an electron transfer term to them. So we're talking about the promotion of electron transfer, the promotion of oxidation and reduction. And we've started thinking about anti-oxidoreductases that people ignore. They're still, in my view, oxidoreductases that are working to suppress electron transfer. And we don't know how many of those there are, but we're going to start looking at how many enzymes there are that have metals that nearly do oxidoreductase action that are totally suppressed in their action. And if they were not, then dangerous. So this range, this is not a clock, but the oxidoreductases cover an enormous range of time. Femtoseconds, when the sun first hits photosynthetic systems, the first step is, the second step is not quite a picosecond. Very, very fast indeed. Then at the other end, days. These are the ones that are suppressed that we are now actually getting interested in. Well, what are they? Up here, primary photochemistry, respiration, and all your metabolic oxidation and reduction. Redox suppression, light harvesting. If electrons move between those green things on trees, there'll be no life on Earth. Would you agree with that? I agree. Good. And every time you breathed, your myoglobin, hemoglobin, actually finally put an electron on the oxygen, we wouldn't be here. And it's called myoglobinemia. Not many people get it because it's suppressed. So we're very interested in these things that don't do electron transfer. So let's have a look at the hydride transfer just to get by it for completion. It's the work of Eyring from the 30s, and it's a large number of enzymes that are more or less classical. If we look close to the Krebs cycle, you can find plenty of them. And they all bind, they might have NADH or flavin, and they transfer a hydride element from one molecule to the other very close. So we've looked about 5 years ago. We stopped looking, but we're about to start again, looking at the statistics of distance, which is the easiest thing to measure in hydride operating oxidoreductases. 87% of structures, you find the gap between hydride donor and hydride acceptor within 5 angstroms. That's almost van der Waals contact. There are also others lying around at longer distances. Those tell you don't believe structures all the time, because these are the things that are you can very easily find that they have to move to get the 2 reactants together. So this transfer of hydride is a very close-hauled action. Same for flavins. So this falls in classical biochemical theory, going back to Arrhenius, a prefactor, and then an activation energy, and over KT or RT. So this is what Eyring did to this. He brought in KT over H, which is a frequency, 10 to the 13th, and he got us going on this kind of picture of enzyme action. So this is a piece of genius by Eyring to pick this number, an infrared frequency, for governing the reaction rate. Kappa A was just the transmission coefficient between 0 and 1, and for the most part, it's close to 1. And when a reaction is occurring from one coordinate to another, the chance of it going over, especially if it's going downhill, will be close to 1. So recently, work done at this institution, actually, has put a quantum cast on this work. Sharon Hammes-Schiffer, and following in the work of Judith Klinman, to say that there is some quantized step in this reaction, which I'm not going to go into here, but I just remind you that it's quantum mechanics, hydride transfer at a very close distance. The hydride may tunnel a fraction of an angstrom. So I'm more interested in electron transfer here, and it's a very different story. This is where Rudy Marcus comes in, and looking at the 4% of electron transfer oxidoreductases, most of them that have provided information about electron tunneling theory in practice in biology have come from photosynthetic systems, electron transfer across membranes, charge separation, oxygen evolving, CO2 fixation. Here. So I'm going to talk about these not in detail, but in how they transfer electrons throughout the talk. So if you look at the structures of photosynthetic reaction centers, they're there simply to separate charge, the green lines here, charge across a membrane. This is 50 angstroms of distance, so electrons have to move quite a long way. Going from one side of the membrane to the other, they charge it up. And the structures, ever since 1985, I think, the first structure of a membrane protein was one of these kind, and it changed the world as far as applying biological tunneling theory to biology. So if you look at, so these are just bacteria, harmless, very useful in the lab, and as you can see, green, and then there's back reactions that are bad, charge separation. They're suppressed in a different kind of way, in fact, due to the theory. Here's a more important one that involves oxygen. This is chemistry. Everything here is electron tunneling. And again, the back reactions occur. And these reactions occur at helium temperatures. They're activationless. And this was discovered in 1960 by my old boss, Britton Chance. And he was the first person to put his finger on biology performing visible quantum mechanics. So these are now, the structures are well known. This is one that John Goldbeck has worked on. It's a partner to the oxygen evolving structure. Light activates it, and electrons can actually go by two pathways across the membrane, ultimately to fix CO2 down here. There's a hydride transfer here. I missed out a flavin. And so, but the point is, these are the structures and for which there is quite a lot known about distance and action. So if you look at the mitochondrion, same kind of picture, no light, but electron transfer is the thing that converts your breathing into charge transfer across the membrane and generation of ATP. So I just show you a few more of these structures. Hemes, copper, and cytochrome C stuck to it. Here's hydrogenase. There's a line of iron-sulfur clusters going to an iron catalytic center that converts hydrogen to protons and vice versa. But the point is, there's a lead-in of iron-sulfur clusters. Nitrogenase, same thing. This is what makes all our nitrogen fixed for agriculture. And there's a lot, obviously it's a very important enzyme. But electron tunneling occurs in it. Another gas exchange, nitrogen fixation, denitrification enzyme. Again, a line of cofactors and gaps between them. Flavin, iron-sulfur cluster, pterin, manganese, molybdenum. Okay, so here's another one. And again, a CO2 fixer as well as an emitter. So how do we measure the distances between these cofactors? Well, we've done a lot of work, which I'm not going to go into here. You can ask about where the electron starts to tunnel from one cofactor to another. And these blobs show you what we've identified as the edge of the cofactor. The electron whizzes around inside these frames, but the moment it steps out to transfer to another cofactor, it's tunneling. It's in the protein. The protein is a good insulator. 50 angstroms of protein will stop an electron for the age of the universe. Proteins are a very good insulator. So if electrons are going to go from A to B through a protein, it's tunneling. So I just make one point here that the edge of the heme is a little contentious. We think it's nailed, but other labs, Harry Gray in particular, and a theorist, David Brereton, also, think that tunneling starts from the metal. We just think it gets a free ride to the edge through a conjugated network. I'm not going to go any further in that. You can ask me at the end. So distances are very important, as I'm going to show you. Distance. Lots of structures. And we got fed up in 2005. We've had enough. But we're now going to have another go because there's so many more fabulous structures doing tunneling out there. So if we look at all the distances of what I've just shown you, inter-cofactor distances, and stick them on our distance plot histogram, it's an amazing result. By the time you get to 14 angstroms, there's no more tunneling done. So it's a block, not a Gaussian or anything fancy. This is just a statistical finding that you'll get tunneling over this kind of distance, 13, 14 angstroms. So we're going to find out what that allows in tunneling terms. But there's no doubt, they're practically all within 14 angstrom distances. So Rudy, who's still alive, still going strong, he got a Nobel Prize for this theory in 1991, and it was a classical theory, actually, they got the prize for. And he wasn't interested in the distances or the electronic coupling between donor and acceptor. He focused on the free energy between a donor and an acceptor, redox potential difference between donor and acceptor. So the reaction, going back to Eyring, wasn't like this at all. It was different. And in fact, he and Eyring actually did a paper in 1955. This was a year after Marcus proposed his Marcus theory, which I'll show you in a minute. So what Eyring said with regard to his own expression, which is up here, there's Ka, kappa A, rate, Kt over Planck's constant, and then the free energy term. He said that if tunneling occurs as well with this chemical term, then you're going to have to multiply this by K kappa E for the electron. And that needs to be included in it. and it would be a description of the probability of an electron penetrating a barrier. And so you'd multiply that by this 10 to the 13th to find out what the rate would be. For tunneling, kappa A will vanish, as you'll see. But this is finding this paper was great to show that these two giants of the world actually work together, and came out with something that focused on, admittedly, the electronic part of the reaction. But the probability of tunneling is minute. And so instead of having a big gap between the coordinates here, you can say there's a little hole. A minute probability, if the coordinates get to this point by dynamic action, there's some small probability of the reaction occurring, electron tunneling. So kappa A turns into kappa E, which is very, very small. How small? Well, Marcus didn't care. He was only interested in what this could allow him to learn from this exchange of the nuclear coordinates from a reactant to product. This would be going like this many times, and it would be governed by how many, what the position of this crossing point would be. That's the basis of his theory. So he was interested in activation energies and what the shape of the rate would be governed by free energy, holding this constant. So this was his experiment, I suppose, in his head. So you have a nuclear displacement, and whatever the displacement is from reactant to product, this point here is the point where it's over the product position, nuclear position. That's called lambda. The crossing point is the thermal action of getting up to that point. The free energy is the drop. So what he told everybody who was experimental to do was change the free energy and measure the rate constant. So this is a point here in the delta G positive region, which I'll come back to later. So he said change the free energy, but hold everything else constant. Okay, and this is what he predicted. And this is now his region of genius, in my view. He said that eventually you'll get to a free energy, a driving force, that will slow down because you've gone up the back side of the parabola. So now it's harder for the system to achieve this particular point where tunneling can occur. So he put a line through this and changed the expression. It becomes parabolic in an ideal approximation with a KT there as well. So this is his famous reorganization energy, typically hundreds of millivolts, large. So this point here would be activationless according to his classical expression. So this is what he did, and he got a Nobel Prize for it. And it was for this, I think, this inverted region was an act of complete genius, recognizing and saying it, that with such a large driving force, it would actually get slower. So you could describe the reaction coordinates in this way. That is what he did. So what about experimental tests of this theory? He put the theory down when he was at Brooklyn College in 1954, and it took till 1984 for anybody to confirm it. And when it came, it was done by John Miller, who's still working at Brookhaven. And he did it in glass. He had mixed donors and acceptors together, flashed it with a pulse radiolysis, and measured the rates between them all and deconvoluted it and got this fabulous parabola. Except he did it at nitrogen temperatures, liquid nitrogen temperatures, and proved that it was quantized. This was occurring at very cold temperatures, and in order to explain this shape, he had to include a vibrational mode, which Marcus only dealt with classical modes, liquid motion. So, nevertheless, I always thought he should have got the prize with him, but he didn't. You know, politics, I guess. But anyway, this was the first demo of Marcus theory. So what about biology? Well, lots of opportunity here. We knew already that these things work at helium temperatures. So this is a photosynthetic reaction center again. Chlorophylls, chlorophyll, pheophytin, that's chlorophyll without a magnesium, and 2 quinones. And so light comes in, excites the chlorophyll, and then it hops down. The driving force drives the electron across the membrane. Every step, there's a chance of coming back. The coming backness is always a lot slower than the forward going. And that's because these free energies are in the inverted region of Marcus parabolas, which I'll show you in a minute. So this work was done by Marilyn Gunner, and it took from 1980 to 1989. She was doing her thesis. She said, "Do not stop me." And she actually stripped the reaction center of everything except the stuff that mattered. So light activation, electrons would go this way and then come back. So you could take the quinone out and put other quinones back. So she became a pharmacologist. Every quinone had to go back into the binding site exactly the same as its predecessor. And so we looked at hundreds of them. But eventually, and then measured the potential, this position of this line with respect to the others, up and down, changing the driving force. And every one, free energy was measured independently. So she repeated, so with this, she could change the free energy of 2 reactions, this and that, and did. So here are her results. She didn't do a Marcus free energy rate plot just at room temperature. She went down to 14K and measured the rates from here to here. Natural rate is 200 picoseconds, so this is very fast reactions. And she got these results. They don't look fabulous. There's a lot of errors. But the only fits, and I'll go into more detail in a minute, the only fits required a quantized fit. So that was the foot in the door for this one. Difficult to measure, but okay. It got the ball rolling. Here's another one, easier to measure. There are 2 families of curves on this graph, from 113K to 5K. So data up here, 35K, 14.5. And how does the Marcus line fit? Well, I can tell you, very badly. So, which we were happy about, of course. So at absolute 0, this would be a delta function. So because his theory is classical, of course it goes narrower and narrower and narrower. It's classical modes. They get colder and colder and go slower and slower. I remember telling Marcus that this was pretty good because this point was 10 to the 42 times too fast measured to fit his line. Okay, so there's no doubt that electron tunneling in biology is quantized. So then fitting other people's work, Joshua Jortner had a single oscillator like John Miller's, and it broadens it out, and of course it becomes temperature independent if the oscillator strength is frozen, is higher than the energy of the temperature, given by the temperature. A fancier one by Levitch with 2 modes, didn't fit very well. It's a monstrous equation that goes on forever. That's not so good. Marilyn's own fit was this, with multiple oscillators, and that fit very comfortably. So we're starting to try to determine whether the vibrational modes were selected by nature, and the answer is definitely not. But nevertheless, we entertained it. In the end, we went to John Hopfield, who was a theorist at Princeton and then Caltech, and now he's back at Princeton. He had a very nice theory that just fitted the results. It wasn't particularly brand new physics, but it made this fit that was pretty good with just one vibrational mode, a Gaussian distribution of them. So this is what we've stuck with for the last 20 years or so. So, what did hot field do to Marcus? Well, at room temperature, at physiological temperature, is not that much. It broadens the parabola. And if you're wondering where kappa E went, it became beta times a distance. So I've not tracked you right into there, but that's where it is. So it's still multiplying the kt over h, 10 to the 13th possibility here. So this is Marcus's equation that he ended up with, dealing with the electronic term as well. We boiled down all these bits and pieces to just into this expression, to the log 10, so you could separate the different terms and do it. You can do calculations in your head. So this prefactor here is all this stuff rolled together for physiological, biological temperatures. Hot fields step, all that work, change this to 3.1 instead of 4.2. That's it. So log, the rate constant, is it 10 to the 13th? We don't know. The kappa E equivalent, and then the nuclear term. So now we can say for sure that in biology, no doubt, nuclei are quantized as well as the electron transfer itself. We think that it's likely to have many vibrational modes, but we're saying definitely not one of them was naturally selected. It's not a radio or anything like that. It's just a generic property of protein. And Darwinian evolution did not notice that it could have tuned. So the best description, we think, is this, a Gaussian distribution around that wavelength. So this is our equation that we've used now for quite a long time, since 1999. So I'm now going to, having got this, I'm going to take you through this so we can put some numbers in here for beta and K at van der Waals contact, it should be. So what about this electronic term? Has the protein been selected to promote good electron tunneling and suppress tunneling that is not wanted, deleterious or unproductive? That's a question that I will answer now. Some people, I should just tell you, for keep it honest, some people still believe that nature has minutely developed the protein to drive electron transfer in the right direction at the right speed. But you can make your own mind up. So how do we get at this? Well, it's not that difficult, actually. If lambda equals delta G, minus delta G plus lambda, this becomes 0. So we can just look at this. So if we compare rates between one electron transfer system and a totally different other, then we're comparing apples with apples if we all look at the rate seen at the top of the curve. So we're addressing whether this is going to turn out to be airings 10 to the 13th per second, and what is going to be the difference, the beta value, which is obviously the slope of the fall-off of rate with increasing distance. The 3.6 here is we subtract the van der Waals radii from the distance. So these are the questions we've asked. How do you get at it? Well, you use a lot of people's work, and there really has been some fabulous inventive work down the years to plot free energy versus rate constant. Marilyn got it going, but then it followed, 10 years later, more, something very, very fast indeed. The free energy was changed by mutagenesis. So you get a rate at lambda, 3 picoseconds here, very fast. Heroic work by Shigeru Ito and Iwaki. Again, taking a quinone out of photosystem 1 and replacing it with many others. And it took them a long time, and they also worked at low temperature. This is their results. The scatter. These are heroic experiments. Every experiment is a separate preparation. And this is the native rate, and then you see the fall off as you go into the endergonic region and fall off in the Marcus inverted region. Amazing results. And in fact, you can see the action here. Nature has picked, in this case, to work at the top of the peak here, this star, and the back reaction is so big it's pushed over here, and it's very slow. So this is a ratchet, click, click, forward, all driven by Marcus theory. So this is perhaps the most important aspect of Marcus theory. Without it, no photosynthesis. Without the inverted region, no photosynthesis, I would say. Anyway, many more have been characterized in this way in different bacteria, and we can plot them on a distance rate plot now. So the green guys are good electron transfer, physiologically beneficial. The red ones are points taken for back reactions, decay, charge recombination. And as you can see, they line up very nicely. And if you start doing a best fit for it, this is the line. And slope is 0.6, and it's not a bad fit at all. And the great thing about it is it crosses the van der Waals contact line at 10 to the 13th. And we were very excited about this, because this isn't a long extrapolation at all. And the line goes down 12 orders of magnitude of change. There aren't many theories that are true from over 12 orders of magnitude. And I mentioned it to Marcus. I said, we've just shown that K/T over H actually appears in photosynthesis. And he said, Les, that's theory. That was it. He wasn't impressed. So this line that first appeared actually in 1992 hasn't changed since then. And points have been put on it. So this is sparsely populated line, we admit. So nevertheless, we now have a number for this. And if this is true, the slope is 0.6. So nevertheless, what it tells you, apart from this single theory for 12 orders of magnitude of time, the productive numbers and the unproductive transfers fit on the same line. That means that the protein as a tunneling medium is the same for them both. There's nothing done to make the green guys high of the line and the red guys below the line. So this rules out the idea that nature has selected, backbones and phenylalanines in a row, pi ways. They're still popular in certain people's minds, but this is the first step to removing that. It's not over. Harry Gray has done a lot of work with, this is now looking for more points. He did work in the 80s as well. Ruthinated heme and other things, iron sulfur clusters and copper proteins. And he did the right thing. And in recent times, there's been theory to really nail the distances, which was lacking initially because there were no structures. These things waggle. But Harry and Jay Winkler did beautiful Marcus plots on this system that's non-physiological. So this is a cluster of their points. This is postmodernist science, because they had a spectrometer that could measure from 10 to the 8th to 10 to the 1. so all their data is in this region. How does it fit? It's fabulous. So these are not physiological, and they fit on the same line. Now, Harry has a different placement of some of these points, but you can ask me about it later. So this is our expression now. This is our little group, this is our description, our guideline for measuring tunneling in proteins. It's based on rock-bottom quantum mechanical theory, rendered down to something useful to biologists. Okay, so what about endergonic tunneling? I've been showing you this positive delta G here. Well, it's a problem, because if it's quantized, this can't go. This cannot carry on as a parabola into this positive delta G region. However, so what we've done is something that might be clunky mathematically, but it still works. And that is, we just add a Boltzmann term instead. And so there's a little sharp curve turn there in the line, but for endergonic reactions, we just add a Boltzmann term to this expression. It would be divided by Kt at different temperatures, but this is for physiological temperatures. And everything's reversible. We're not breaking any laws of thermodynamics here. But it's a little clunky, but it works. And it allows us, nobody else does this, it allowed us to calculate rates of tunneling that are net uphill. Not for the tunneling, but for the actual system. So just to look at variances of lambdas in biology, they go from 0.2 to 1.3 volts. They're large numbers. And typically, we use the rule of thumb, 400 millivolts for inside nonpolar proteins, 0.2 to 0.4. These are typical numbers for hydrophilic proteins, and then very polar media, watery, 1.3 EVs. So this is a number you can sort of use reasonably reliably when you're using our equations. Looking at this overall, there's one big thing, that small lambdas do make tunneling that are driven by zero driving force, which is what biology does all the time. It's very cheap. Most reactions in biology are either slightly downhill. In the end, it's downhill. But they have very small numbers. And therefore, a small lambda would help push up the number and also push up the reaction rate if it's going uphill. Okay. And it's also for photosynthesis. A small lambda would help the back reaction be in this inverted region. So one more thing here. The 0.6 slope on this reaction, does it have any roots? Is it purely empirical without any history? We think it does have a history. So here's our line again with a beta value. I've put it back to beta and see where it comes from. So there's been a lot of work. I'm not putting the names of people on this at this present moment. I've done electron tunneling from a donor to acceptor in a variety of liquids, ices, structures. And they're quite well characterized. So if you have a donor and an acceptor with a conjugated link between the two, like a carotenoid, then it's almost like an electric wire, not quite. But so you can tunnel a long way very fast. If you get rid of the continuous orbital overlaps, like you can put partially conjugated phenols, there's a higher barrier, more of a penalty. Sigma bonds, CH2, CH2, more steep. Broken solvent in solution. This is the work that John Miller first did. Just solvent. It's difficult for electrons to tunnel through disjointed media. Water, Harry Gray has done this, and the slope is steeper still. Electrons don't find it easy to tunnel through there. And vacuum, 1.2. Well, that's electronics. That's why you use electronics in vacuum tubes. So tunneling is very steeply falling off. So where does this lie? So let's look at biology. It turns out, if you do enough of them, you get a distribution of packing density. Molecules packed in a protein. Actually, the protein is only 75% substance. Phenylalanines, methyls, and whatnot. So the remainder, we've contended, is vacuum. There's nothing there. And so if you treat that this way and look at this again and split our beta value to include a packing density, then we maintain that this is a product of sigma bonds and vacuum, of which there's 75% of this and 25% of that. And if you plug that into here, you will get 0.6. And so that's where we stand right now. It's not proven, but it's well supported. And there are very few deviations from the 75% packed. But nevertheless, we use 0.6 in our expression. So how good are these? How good is this expression overall? Well, the answer is pretty good. You can include in your test rho, variation of packing, or not. And the data of calculated rate versus experimentally well-known rates, they're identical, or statistically so. And the standard deviation of any of these points is something like 6 or 7-fold in error, because it's a log-based thing. So 6 or 7-fold error in a calculated rate in biology, it's not so bad, okay? A distance variance of half an angstrom in a crystal structure will give you the same kind of variation, okay? So we think it's as good as it can be. We keep this in mind, this part of the electronic term, but generally we just drop it to 0.6 as the prefactor. So these are our equations developed over the last 20 years or so, and they do approximate quantized descriptions, electronic and nuclear. They are proven accurate to within experimental uncertainty, and we think they capture the essence of natural electron transfer. So let's try them out on something, putting these expressions to work. So now we can look at our distance plot of all those tunneling oxidoreductases of nature, and we can put some numbers on what the distances mean. And the answer is, at maximum rate, you can see that from van der Waals contact to 15 angstroms, it's all faster than nanoseconds. This is biology doing nanosecond to peak femtosecond reactions. So at free energy of 0, which is typical, and even uphill, which I'm going to talk about next, with a big lambda, generous lambda, you're in the micro to nanosecond range, even 10 to the 10th at touching. So this is an easy kind of piece of engineering to get something far faster than biological oxidation reduction enzymes, which are typically milliseconds. So there's a lot of room to play here. So these are my reaction sensors I've been showing you. And let's look at, so these are the way these distances play out. The ones that matter, the ones that capture photons and initially separate charge, well, they're very fast indeed, and the distances are very short. Makes sense, right? The others don't matter. There's no statistical sense to where the other distances lie here. So let's look at some reactions now. This peculiar reaction center has 4 hemes on it, and a 5th binds to the end one. 1, 2, 3, 4. These feed electrons into the chlorophyll, okay? But they're very peculiar, and people have wondered, how can this work? So we all, this is the free energy course of electrons hopping from here to here to here. There are these gigantic roller coaster uphills. This is 300 or 400 millivolts uphill. That's 8 kilocalories or something like that. So why did nature arrange its chains like this? If you look in early textbooks, you'll see electrons are always hopping downhill. They have to be very carefully handled. But clearly, what is this for? So we can now calculate rates with the free energies and putting in a lambda. We do all the differential equations, join everything to everything else, completely reversible. And these are the results we get for an electron going from here to the chlorophyll. From here to the chlorophyll, it's 10 to the 4th per second, 100 microseconds. Fast. Can easily keep up with photosynthesis. How much? It's like 1 photon per second. Pardon? Yeah, 1,000 per second. So it can keep up with the light coming in. And each, just going over from here to here, it's 10 to the 5th. So electrons, aided by the nuclei, can go over hills. And this is a revelation to people who are been brought up to the way biology is designed very carefully and everything organized to be optimal. So let's have another look and see how nature uses it in this enzyme. So it oxidizes quinol and reduces nitrate to nitrite. So the arrangement of the distances in that enzyme, plus many of its neighbors, I think 4 different ones here. Again, the catalytic distances, short, so that the intrinsic tunneling rate to do chemistry, banging on the chemistry door, is extremely fast, approaching 10 to the 13th. The others don't matter. They can keep up. It's still fast. So how does this work? Well, this is another amazing roller coaster. So this actually, an electron starting here, I've thrown the substrates away. If an electron starts here, it goes all the way over here and finally goes to the molybdenum and the substrate. So what you would guess how long it would take one electron to get here to there, going up 600 millivolts. That's 12 kilocalories of uphill, much bigger than KT. Well, the answer is, we can calculate this now, because we have our tunneling expressions. And the answer is 28 seconds, which isn't bad, actually. So from here to here, including a thermally activated uphill step, down here. What happens if we put 2 in? The answer is 1.6 seconds. What if 3? 37 milliseconds. 4? 2 milliseconds. And now you're talking, because with the quinone on it, the quinone can easily put 4 electrons into this manifold of this chain, in which case, when there's quinol around, the electrons will flow, not before. So this is a regulation for, if there is substrate, then electrons will get from here to the nitrate to reduce it in milliseconds. And the kcat is 10 to the 3rd per second. This probably is close to kcat. But this is a nice kind of regulation using pretty extreme numbers of free energy up and down hills. Hydrogenase, the same. It's got a big trough in the middle, and yet it's one of the fastest enzymes in the world. The kcat of these is something like 40,000 per second. It can easily keep up with that. So common in nature. So looking at photosystem 1, I'm just going to show you that with all the up and down hills, we can do a complete analysis of this and get a complete readout of all the different steps of this continuous sequential action, and describing both pathways in the right rates and the right proportions. So I hope I've given you a decent picture of a fairly robust engineering of electron tunneling in biology by electron transfer. So there's a lot of others that do hydride transfer and electron transfer, and I'll take you through that quicker because it's the same story. In mitochondria, you've got the respiratory chain, the structures of them all now, the mechanism of every energy transducing machine in respiration is totally different. It was not one movement to get these things designed. So this has a hydride transfer at the NADH step with flavin, and then there's a whole chain of iron-sulfur clusters ending up in a quinone. Succinate dehydrogenase, a shorter chain, ending up with hemes and a quinone. BC1 complex, it's a twin, and I'm going to come back to the twin in a minute. 4 hemes in it and an iron-sulfur cluster, and 2 quinone sites. Twin, it's a dimer. You can't tell the difference between one and another, and I'll come back to that. And then cytochrome oxidase, which I'm not going to talk about very much at all, reducing oxygen. So if we look at this, the electrons, for years and years, people have puzzled about how the electrons can go from here to here. It's something like 80 angstroms, a long chain. And the quinone at the end never does hydride transfer. It's 2 electrons, one at a time, and protons. Just telling you that. Here is the redox profile as we had it some years ago, a couple of years ago. This has changed. I'll show you in a minute. But we can calculate the transit time on this system from here all the way to here. So I have a, let's see. So here is the time progress of transfer of just one electron from here right across. And you can see it's all over in the required 10 to the 5th per second time scale. So these things also work very quickly indeed. And recent work by Judy Hurst in Cambridge. She's remeasured the potentials of these hemes, for it is her institute that had the structure of the complex 1, and has also found that the up and downness has become far more extreme than we had it. She's done some theory work on whether it actually manifests itself as this kind of up and roller coaster, correcting for what is it? Epsilon, forgot. Dielectric constant, sorry. Right. So that's up in the air right now. But again, even though her profiles of transfer from here to here are different, nevertheless, it gets there between 3 and 4, 10 to the 3rd, 10 to the 4th per second. She's saying it's too slow. We're saying if you put 2 electrons in here, it will be fast. It will act more like a wire. So it's the same story for complex 2 here, which has the same kind of pattern, a roller coaster, up and down. It still gets there in a rate that is on time. I'm not going to bother with that. Let's move to this one, complex 3. It's the same story. 2 electrons come in from the quinone. One goes one way, one goes the other. This was the cause of another Nobel Prize, and that's Peter Mitchell for his chemiosmotic hypothesis. This put the nail right into it and got it. But the mechanism's still outstanding, and whether there is inter-monomer electron transfer is a question. But here again, the electron, the reaction is over in milliseconds, in plenty of good time for the reaction to occur and keep up. So what I'm showing you here is an extension of what I showed you in the last section, and I can now perhaps emphasize a little more what the basis of chain engineering is. Basically, putting cofactors into between one donor and acceptor, equipping it with others, essentially changes an extremely strong exponential fall-off of rate with distance. A 32 angstrom distance would be towards the age of the universe, I think. Certainly a very long time indeed. You put one cofactor in the middle, it's 10 angstroms long, it becomes a microsecond from 3 hours. So sorry, that's 3 hours, not, I'm exaggerating. Okay. 3 hours, age of the universe, no problem. Okay. I think it's 52 that's the age of the universe. You can work it out. All right, 3 hours, too long. One cofactor, a microsecond, 2.5 nanoseconds, 3. So it essentially linearizes. This changes the rate fall off with distance from exponential to linear. So if we made this twice as long, it would be 2 picoseconds. Amazing. One thing that nature can tell the difference between is geometric and arithmetic relationship. This is just so simple to engineer. So the close cofactors make the whole system very much more tolerant to bothering with cofactor variance of structures, lambdas, and the free energy between them. And if you have them adjusted to the right distance, you can very easily get rates that are far greater than diffusion or chemical catalysis, readily meet photochemical requirements, and the scale of the barriers encountered can easily, and evidently do, can keep up with the chemistry of multi-electron catalysis or hydride transfer. So such chains, they promote electrons on enormous scales, distances, times to picoseconds, easily obtained, spanning membranes and joining distant catalytic sites. Some of the reasons for chains are to keep one catalytic site from attacking the other, for example. And there's another example I'll show you in a minute. So an enormous regulatory power. This is pure electronics to control electron transfer, slow it, speed it, as well as providing a sink for multi-site electrons or holes, and with rapid succession of electrons for chemistry. So I might be beating a dead horse here, but I'll just go through a summary. Distance through protein is dominant, overwhelming, but totally controllable easily. Free energy negative, downhill, driving force is important, but not for biological speed, merely to tilt the reaction in the right direction. And of course it's cheap. It doesn't cost very much. Uphill steps, well, short circuits in photosynthesis and regulation that I've shown you in chains. And also uphill for tunneling frequency, banging of on the electronic door of chemical catalysis is aided by short distances. And lambda, all that trouble for lambda, it's a fairly dull parameter. We think it's critical in early events in photosynthesis for visible light. But when it's small, not difficult to get, hydrophobic interiors of proteins will do it. It will also make it go fast forward, but also allow, in photosynthesis, to get charge recombination slowed by having an enormous recombinating delta G. But you can guess it, and it's probably going to be good enough. So beta, we just think to start off with, a 0.6R would be good enough, but watch out. There may be a pathway one day found. H bar omega, generic at this present moment. So that brings electron tunneling from quantum physics and chemistry up to biology. So let's now have an analytical look and have a look at unknown, decent questions to try and answer with our theory. So the BC1 complex, everybody's known for Dunkish years that it has two identical subunits. The two subunits, one doesn't affect the other. The color, the spectra of everything are identical. I know because I've worked on it. I've worked on it since 1972. And I retired my grant that I had on it last year. And I gave up. So what you're going to see here is my last experiment. So this is the question. It's not an earth-shattering question. Although at the end it might be saving our lives. Everything we do must be important. So, especially for NIH. Okay, so question. Can an electron go from subunit 1 to subunit 2? Well, this kind of gap between these identical subunits appears across the phyla, microorganisms, plants, mitochondria. So here are the distances between the cofactors, and the gap is on the edge of our 14 angstrom statistics, 13.9. Well, we can calculate the rate because we know how to do it. Lambda, 0.9, we're guessing that. Delta G, 0, they're identical. And it comes out at 0.1 milliseconds. Oh, it's not bad, 10 to the 4th per sec. So it keeps up with the enzyme action of this, which is a millisecond. Okay, so it could occur, right? So Artur Asichka, who was in my lab, but he's now in Krakow on his own, doing very well, he got around this problem of two genes expressing two separate identical subunits. He'd stitched them together and made it into one protein. Brilliant. And it did work. It's fragile, but it works. So now, if you have one protein, you can actually do asymmetric mutagenesis, which was the big problem. Every time you tickle this one, you tickle the other side, identically. So this is a schematic of the reactions that I showed you before. And the question is, how do you get to test whether it goes across? Well, you don't have to think too hard. You can block that one and not the other. You can block this and not the other. You can block them both. You can do that. But if you do that, that's the test. So if you can activate this side, does the electron end up on that B? So block the exits totally. There's lots of inhibitors to this protein. Look at this B. You can tell I've given up this grant. Okay, here's the answer. So quinones come in, I've not bothered, and then cytochrome C is oxidized, so it's now activated. And the quinol comes into this site. This separates charge, and yes, it goes across. And it goes across in a millisecond, which isn't bad. So the control is stop the electron getting in here. I'm not bothering you with the details here. It's good enough. Nothing happens. So this is a proof that there is contact between the two subunits. Is that exciting? Well, it is in a way. So now we can say, okay, which way does the electron go? So the electron, it can either go this way or that way now. So from here to here, 1 millisecond, we got 0.1. That's all right for us, guessing. Our calculations within a factor of 10. We like it closer, but that's okay. If an electron gets here, it will choose to go down here in 1 to 10 microseconds. That's measured. And it has a big driving force, moderate anyway, driving force 3 to 4 kilocalories to have it go that way. So you expect the crossing, bridging, to be ignored. So the picture is like this. Comes to this part, and it goes on, 100 to 1. So what good is this? So, well, the answer is not so good. It just means that they could talk to each other if they wanted, but this kind of difference is a big difference. So, we weren't looking in the right place. These things are working machines transferring charges across the membrane. So they don't work without there being a transmembrane electric gradient, electrochemical gradient of protons. So we've done more calculations and to ask this question. So these things are all hooked together in the respiratory chain, and their job is to separate charge, pump protons, separate charge, and the value of the charge difference is 200 millivolts, which is quite a lot of electric field if it's all in electrical terms. The protons go on to make ATP. So how does the BC1 complex feel about this? So these are the catalytic energy coupling sites. Let's have a look at BC1 again and ask again, can it go across? Or would it go across? And here we're looking at it going 100 to 1 in the wrong direction. But we think, what if it came back? Because there's a big driving force for quinone to become a quinol. Dismutation, or the opposite of dismutation. So this could happen. But so we can calculate it. And in other words, 2 subunits, 1 turnover completing the whole reaction sequence. So let's look at that. Well, with no membrane potential, it takes 10 seconds. We can calculate that now. That's no good. 10 seconds. The system won't notice. But if we look again and ask this question with an electric field across it, then the barrier, this is an uphill reaction, the big distances. Under the influence of field, the uphill step will get smaller and smaller. So we calculated that under different field strengths, 0.1, 0.2 volts. And so what you see is that when it's operating under transmembrane electric gradient, it becomes milliseconds to microseconds. So when it's doing the job, instead of being dragged out into the lab, stripped of its membrane, it works. So this is, why? Well, there's a very good reason. That quinone radicals are removed, and this would suppress superoxide generation, which, of course, is harmful to us. It could be redundancy, and it could be other things, but maybe none of the above. But this is a very likely candidate because this particular complex is generating superoxide, peroxide, and hydroxyl radicals all the time, especially during reperfusion after operations. So, and it's slowly, leaking out these noxious chemicals which cause us all to get old. And certainly it's a very serious issue in the operating theatre. Okay, so even though it doesn't change the mechanism or the stoichiometry, we can't ignore the fact that this is now an operating pair. That's the end of that. So I'm just going to show you something that's emerging now before I end. There are now, this is now chains extraordinary. There are chains being found in bacteria that actually come out of the bacteria, stick onto oxidized ore, iron ore, or reduced ore, and essentially suck in oxidizing equivalents or pump out electrons. And there they have 10 hemes. They're all heme C types, they're covalently linked. There's a lot of theory now going on by Jochen Blumberger in London. And it's the same story, up and down hills. These are his pictures. It's all quantum mechanics and molecular dynamics, but it's coming out to the same as everything I've told you. I can show you more. The rates are all in this range, 10 to the 5th to 9th. So it's the same story. There's one with 10 hemes attached. Here they all are. So, so these are everywhere. There's this one which has multiple catalytic activities in it. And I don't know how many there are, 20 or 18 hemes, all stuck together. They're wires. They're so close that they act like wires. So these chains, they're everywhere. This is not just a small item of academic interest. So this kind of molecular electronics is really, I think, a very exciting option to work on. Okay, and indeed, it's a very exciting option to work on, making them ourselves with homemade protein. So I'll end by very shortly just showing you where we can go now with what we have. So natural oxidoreductases, these kind of ranges, man-made. And when I'm saying man-made, I mean no mimicry, starting from scratch. And I'm only going to give you just a picture of this. So we call them maquettes. Maquettes are models, working models. Architects used to make them before computers to show the client a picture of their building. A working model. So that's what we're making. So we make them based on basic principles of protein folding, alpha helical folding, beta sheets. These are very well known, understood enough to do it, to make it. And I hope you agree that we've got a good foot in the door for understanding electron transfer engineering. So we design structures and functions that are completely transparent, no scratching head, minimal structures, man-made, easily made. We're trying to test in an in vitro environment to repeat what we've seen in biology. And as I told you earlier, we would love to integrate these proteins into photosynthetic systems to really do gene manipulation to make fuels, hydrogen particularly, or to correct electronic problems in humans. So those are our dreams. The picture is shown here. It all started with alpha helices. That's what these cylinders are. Started in the 80s by Lynn Reagan and Bill DeGrado. Since then, we've got a string of these things now. We can make a very large number of different proteins. And we've now got a 16 alpha helical structure coming. And in membranes. And these frames, we know what every amino acid in them is doing. And we can equip them with all the cofactors I've been talking to you about today. Well, if you multiply this by that, you get a lot of possibilities. We all can do arithmetic. Enormous opportunity. And these are very stable. I'll just give you a hint later. We've already got them on surfaces. They are extraordinarily stable. I'll show you later. You can take them up to above boiling point, and they still hang together, even with the cofactors in. They make beautiful films, print, and so on. They go into membranes. That's struggling a little more because it's difficult to express them. All these are expressed, so it's very inexpensive. And our goal is this, back to nature. So the way we do it, we play in a sandbox, and we don't talk to anybody. And we play around with these different maquette frames on cofactors, borrowed from nature, admittedly. Substrate goes in, and we have some standards. They're totally different from the kind of standards I grew up with, studying natural proteins. They're monodispersed, stable helix topology, malleable interior. We're not looking at exact structures. We like them a little flexible first. Independent construction domains. These have all been tested. Cofactor binding, very rapid, very tight, picomolar often. In vitro properties, step-by-step changes we can actually progressively develop. We don't do anything that's not biocompatible because of our goal. And we are interested in biogenesis of the whole thing, not just the protein, cofactors and all. And then it goes back to simplification and revision. So what have we done? These 4-helix bundles with 2 hemes in here, I'm just giving you 2 examples, hemes and oxygen. Well, we can bind to these hemes any of these small gas molecules reversibly and attach them. We've formed oxyforms of all these different enzymes. They look exactly like natural ones. We are just starting to get them to work. And anything blue here, we haven't done yet. Okay? But this is where we are. You've got to get the oxygen on first to do chemistry. And that is part of our suppression, in a way. We can control oxygen binding without oxidation. Now we're turning it on. Here, we're suppressing further oxidation because we're interested in making signalers that are transcription factors or blood substitutes. So on the other side, we can make these hemes oxidize water, oxygen, immediately, promotion, creating things like, well, I'll get there, NADH oxidase, neutrophil generation of reactive oxygen species. And we're also interested in making those wires that we are involved with. And we have made diffusional proteins too. So biogenesis, very important. So we've now just managed, it's published, to get one of our man-made proteins to incorporate heme C, dithiolated linked, all in E. coli. And it's beautiful. And it also binds oxygen. There's no such thing in nature, a heme C that binds oxygen. But it could be the beginning of a blood substitute. So heme C biogenesis, it certainly maquettes interface with the biogenic machinery of cells. We can build something in one helix pair and another in the other, and so on and so forth. It has no relationship to any globin fold, and it's a natural model for synthetic blood. It had, when we made it, 14 amino acids. We've actually shrunk it down to 7, being very quick here. 7 amino acids. Every one, we know what they're doing. Hemes bind in astonishingly tight binding, and it behaves very similar to blood oxygen exchange in natural blood. We're working on that. And the other thing is that it's stable to above 100 degrees. It can be autoclaved. Good. So think about that. No bacteria growing in this blood. So it's hyperthermal, autoclavable, and it can be stored indefinitely. And it has double the oxygen capacity. So this is just using the same kind of, thinking as the rest of my talk, but this is prevention, suppression. So on the photo side, we're doing the same kind of thing. We're interested in suppressing electron transfer in light harvesting. And we have maquettes that cover the spectrum, cover the solar spectrum now, and perform light-activated energy transfer. So quick for that, that's suppression. On the other side, we're trying to make a photosystem too that will evolve oxygen and maybe evolve hydrogen one day. Comparing with natural photosystem 2, inside there's these functional active units. This is the size of our maquette compared to a photosystem 2. We can put 4 or 5 different cofactors in. Each cofactor knows where to go. Very simple rules. We now have a crystal structure of a 62 angstrom long homemade protein made with just very few different amino acids. And if you're not impressed by this, you should be. It's 1.4 angstrom resolution. I mean, this brings tears to most X-ray crystallographers to actually to see through the macrocycle. So here's our structure. It's got a heme in instead of QA, a chlorine, which is synthetic. In there is tyrosine and the beginnings of a 4-manganese calcium cluster. It's currently just 2. But nevertheless, we are seeing now for the first time details of our own homemade proteins, and actually we're well on our way to getting this thing fully active. Currently it works from the triplet state. We will fix that by giving this guy a partner. But you see, with it, we do all our electron tunneling modeling as we're moving along with this. So, will it set itself up for expression in natural systems? Well, we're practicing. So we've managed now, we'd love to get photoactive photochemistry in green bacteria, cyanobacteria. Currently, we've got one of our maquettes with 2 zinc porphyrins in there, fused to one of these phycocyanin molecules. It's all expressed in the cell, and we feel there's no reason why we shouldn't eventually insert one of our charge-separating maquettes into the light harvesting system of photosynthetic proteins in algae in particular. So that's the goal. And that's the end of my talk. Thank you.